Let's talk about tissue and organ transplantation in terms of what are the factors that clinicians take into account to prevent transplant rejection. So let's define some terms here. We have the donor, who is giving the organ or tissue or blood, and we have the recipient, who is receiving the organ or tissue or blood. And the main question that clinicians ask here is the organ or tissue histocompatible? Is the recipient receiving a histocompatible organ or tissue? So what are we talking about here? Histocompatibility refers to the notion that the immune system in the recipient, it would be great if the immune system in the recipient identified the donated organ as self, as not something that is foreign to the body. Because, as we've talked about in this course, the function of the immune system is to recognize something that is in the body as non-self, recognition, and then have an effector function and attack that. And so we don't want that in tissue transplantation. We don't want rejection. So we want to minimize the ability of the recipient's immune system to recognize the donor's organs or tissues as foreign or non-self. So we want to get as much histocompatibility as possible so that the recipient's immune system doesn't identify the donor's organ or tissue as non-self. And there are two main sets of factors that we look at when we look at histocompatibility. In the previous video, we talked about the blood group antigens, the ABO antigens. These are glycolipids found on the surface of erythrocytes, also found on the surface of endothelial cells. And if these glycolipids are sufficiently different between the donor and the recipient, then the recipient is going to recognize the donor's blood antigens as non-self, and once it recognizes them as non-self, it will attack them. So during organ transplantation, as well as blood transfusions, we try to make sure that these two are compatible. The donor and the recipient have a compatible set of blood antigens, right? So that's one thing that's considered. The next thing is the MHC complex, so the major histocompatibility complex. These are the molecules found on the surface of cells that are involved in antigen presentation. Now, you'll see these are called major histocompatibility complexes, and that is because they are the primary molecule found on the surface of the cells that are recognized during tissue and organ transplantation. And that's why they're called the MHC, because this is the major thing that determines histocompatibility. So the donor has MHC molecules on the surface of their cells. The recipient has MHC molecules on the surface of their cells. Why are they compatible or incompatible? What do we mean by that? So this requires you to remember that, first off, there are a number of genes that code for the proteins found in the MHC. So when we're talking about MHC class 1 molecules, we're talking about the HLA A, B, and C genes, which code for the alpha chain of MHC class 1. For MHC class 2, that we're talking about the alpha and the beta chain of the MHC class 2 molecule, coded by genes such as HLA DR, DQ, DP, which are not shown here. So the donor has HLA genes, the recipient has HLA genes. So what do we talk about? What do we mean by compatibility? So you have to recall that there are hundreds, if not thousands, of different alleles or allotypes of these genes. So I've given here an example. The donor, let's say, has HLA A version 29, B version 57, C version 03. And again, as of the recording of this video, there are almost 2,000 different HLA-A alleles, over 2,000 HLA-B alleles, 1,500 or so HLA-C alleles. So each of those alleles are going to encode for a slightly different HLA alpha chain that participates in antigen presentation. Now, the individual who's receiving an organ or tissue, they may have different HLA alleles that they inherit. Some may be the same, and this is what we talk about tissue typing, tissue matching. You want to make sure there's a match between the donor and the recipient, as many as possible. So maybe they both share the exact same HLA-A gene, but the HLA-B and C genes, those are different, right? So there's not a match there. So when the donor gives the recipient some cells, those cells might have MHC class 1 or 2 molecules on their surface that are slightly different than the recipient's HLA or MHC molecules. So if there are any non-self HLA molecules or MHC molecules present on the surface of these organs, on the cells of these organs donated to the recipient, then it is possible that the recipient will generate antibodies that will bind these molecules. Because these molecules, they're non-self. I mean, they are just MHC class 1 molecules. Obviously, the recipient has MHC class 1 molecules, but HLA B57 is slightly different than HLA B42. So if that difference is noticed by the immune system might make antibodies to HLA B57. These antibodies are known as alloantibodies, and these antigens are known as alloantigens. So we're not talking about antigens in terms of something coming from a pathogen. Now we're talking about something coming from a donated organ or tissue. And they're referred to as alloantigens because clearly the recipient has MHC molecules on the surface of their normal cells, but they're making antibodies to HLA molecules found on donated tissue. So we call these the alloantigens, and this is an alloreaction. So we want to match as many antigens between the donor and the recipient as possible. These HLA antibodies, or I should say these anti-HLA antibodies, where would they come from? Why would an individual generate anti-HLA antibodies? So there are 3 scenarios we can talk about here. The first is during pregnancy, and this is restricted to females. So I've drawn the genome of a female here, the genes that code for the HLA class 1 and class 2 genes, and I'm not going to list them all out, but obviously A, B, C, DP, DQ, DR. I didn't write DR. I should write DR right there. Oh, yeah. And I've drawn both chromosomes at this point here for the female. In the last slide, we only drew 1 chromosome. So if and when a female gets pregnant, her fetus is going to inherit one of her chromosomes with her HLAs, but also the parental HLAs. So that I've drawn that there in blue. That's the parental HLA haplotype. So the fetus's cells will have both the maternal HLAs and the paternal HLAs. Now, during childbirth, usually some of the fetus's cells enter into the mother's circulatory system due to the separating of the placenta and the bleeding that occurs during childbirth, which means that those fetal cells will enter the maternal blood supply and could interact with the maternal immune system, and the maternal immune system might recognize those HLA molecules that are paternal as foreign or non-self molecules, and then could generate anti-HLA antibodies. So the more often a female is pregnant and gives birth, the higher her chances are that she's making anti-HLA antibodies. So that's a very specific instance where an individual would make anti-HLA antibodies, and again, these would recognize the paternal HLA molecules. The second instance where an individual would be exposed to foreign or non-self HLAs and generate anti-HLA antibodies would be during blood transfusions. Now, during blood transfusions, we've spoken previously about this, erythrocytes do not have MHC class 1 or 2 molecules on their surface. So that's not an issue here. What is an issue is that sometimes during blood transfusions, there are white blood cells or leukocytes in the blood, as well as platelets. Now, it turns out leukocytes, like dendritic cells, monocytes or macrophages, B cells, those have MHC class 1 and class 2 on their surface. Platelets, it turns out, also have MHC class 1 on their surface. So if during a blood transfusion, leukocytes or platelets are given to the recipient, and those HLAs or MHCs are not matched, then those could be recognized by the recipient's immune system as foreign or non-self molecules, and then the recipient's immune system could generate antibodies that would recognize these. So that's a second instance. The third instance would actually be during organ transplantation, during and after organ transplantation. So again, we talk about tissue typing. We talk about matching between donor and the recipients. And when we're talking about this type of matching, what scientists are really talking about are matching the HLA class 1 molecules and the HLA class 2 molecules, at least the polymorphic ones. So HLA A, B, and C, that code for MHC class 1 alpha chain, and the HLA DR, which is the most polymorphic of the MHC class 2 genes. So these are the ones that are most likely to be mismatched, and the ones we would love to match as much as we can. So here we're trying to match a number of genes between donor and the recipient. So let's say a donor comes forward. It could be a family member. It could be, if it's a family member, they're more likely to share the exact same HLA-A alleles, A, B, C, DR alleles, but it could be a non-family member. And so let's say here, the donor and the recipient, they match 4 out of the 8 genes that we're looking at here. So all the genes in black, they match. They have the exact same alleles in those positions. But the brown alleles, those do not match. They are slightly different. The more different they are, the more likely that they're going to be recognized by the recipient's immune system as non-self molecules. So let's say the recipient needs a kidney, and the donor, well, we match 4 out of 8. So the donor's kidneys the donor's kidney is removed and sent into the recipient. Now, there are MHC molecules on the surface of the donor's kidney cells and other cells that you're going to find in the kidney. And those might include those identical MHC molecules between donor and the recipient, as well as the non-identical MHC molecules between the donor and the recipient. And it is those non-identical ones, those mismatched HLAs, that again could be recognized by the immune system, because if they are different enough to be recognized as non-self molecules, they would elicit an immune response. And these antigens, which we call now alloantigens, would generate these alloantibodies, which are anti-HLA antibodies. And they wouldn't recognize the recipient's HLAs, they wouldn't recognize the recipient's MHC class 1 molecules, and they wouldn't recognize the ones that match between the donor and the recipient. They would recognize the ones that do not match between donor and the recipient. So these are 3 instances where an individual would generate anti-HLA antibodies, and these are one of the primary reasons for organ and tissue rejection.